Okay, it looks like it's about time to get started. So thank you all for joining us today for our webinar. My name is Jackie Carville, and I'll be presenting the webinar today. I'm here to go over some of the functionality of DNASTAR's latest protein structure prediction application, NovaFold. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. To ask a question, simply type it into the chat dialog and select Send to Host. I will then answer these questions for you during breaks in the presentation. If you need any assistance or have any questions during the webinar, you can send me a chat message or email our account at webinars@dnastar.com, and we'll have someone assist you. And with that, I'll get started here. So to get started, I'll be giving a little background on the company as well as NovaFold itself, and then getting into the software. So we are a global software company headquartered in Madison, Wisconsin. Our sales team, developers, and support network are all here, so if you have any questions, we have the full support staff to answer you, and then can use your feedback to make our software even better. We were born out of the E. coli lab of Dr. Fred Blattner at the University of Wisconsin in 1984. We built a strong reputation for ourselves during the Sanger sequencing years that continues on today. While still offering these classic sequence assembly and analysis tools, we've also expanded into the realms of next-gen sequencing, supporting all major next-gen sequencing platforms. In addition, we offer software to assist the work of structural biologists, as we're going to discuss more in this webinar today. Now, DNASTAR products have long been established on the desktop computer, but are additionally available on either the Linux cluster or the new DNASTAR cloud. DNASTAR is the leader in citations in peer-reviewed journals. And in looking at this graph, we can see that for the past 28 years, more researchers have cited DNASTAR software in scientific journals than any other sequence analysis software on the market today. Now, DNASTAR began venturing into protein analysis software back in 1998 with the introduction of Protein. Protein was created to mine information from protein sequences, helping to predict and display patterns, secondary structural characteristics, and physiochemical properties of protein sequences. However, in 2011, Protein 3D was introduced. Protein 3D is our application for exploring macromolecular structure, motion, and function. Within Protein 3D, you can use graphical views to visualize your protein structure, employ various prediction methods to evaluate secondary structure characteristics, and easily customize your structure rendering. Now in 2013, we have introduced NovaFold. NovaFold is our new protein structure prediction program. Integrated with Protein 3D, NovaFold features the rich graphical interface of Protein 3D with the high-performance computing of the Amazon cloud. With the purchase of our Lasergene Structural Biology Suite, you will receive Protein, Protein 3D, several other sequence editing and analysis tools, as well as access to NovaFold. Now with the introduction of NovaFold, we have shifted our historical licensing model as a company. Traditionally, all DNASTAR software was purchased as a perpetual license. However, with the introduction of the DNASTAR Cloud in our Lasergene 11 release a few months back, and the introduction of NovaFold, we now offer more flexible licensing options customizable to your particular project. Now, you have the option to set up annual licenses or licenses by month, week, or individual NovaFold structure prediction. Our sales representatives are happy to talk to you about your specific research and set up a license plan that's customized to your particular needs. Now, NovaFold as an application was born out of an ongoing collaboration with Dr. Zhang of the University of Michigan. NovaFold utilizes the Zhang Lab's iTaser algorithms to build accurate, full 3D atomic models of proteins with previously unknown structures. So why this iTaser algorithm for our particular application? Now, to explain this a little better, we'll have to go further into analysis of the CASP competition. Now, this CASP competition is the Critical Assessment of Structure Prediction competition. This is held every 2 years where hundreds of teams test their new structure prediction tools and algorithms against newly solved protein structures. Now, this particular competition entails a blind study over 100 different structure targets, and these predictions are assessed by an independent panel of scientists. Now, this iTaser algorithm out of the Zhang lab of University of Michigan has won the competition for the past 8 years. Now, if we just take a look at these numbers for known sequence entries in GenBank, and then compare them to the number of known protein structures in PDB, we can clearly see a huge discrepancy between these two different data points here. Protein structure prediction programs are so important because they allow us to work with proteins that cannot be or have not yet been crystallized. Now, there are three major structure prediction methods that I'd like to discuss today, and those are homology modeling, threading or fold recognition, and ab initio folding. Now, first, homology modeling is the easiest case of all three methods and uses a solved homologous structure for calculations. This method is usually successful for about a third of all proteins. The threading method is a method used for harder structure prediction problems and will require a similar structural fold to the predicted protein. Now, this method is usually successful for about 50% of all proteins. And finally, ab initio folding is for the most difficult prediction cases, which require intensive calculations on the biophysical principles of the protein. Because of this, ab initio folding works best for small proteins that are about 100 residues in length. Now, these two images below demonstrate the accuracy of this ab initio method. The image on the left was created using the ab initio folding structural prediction method in the iTaser algorithm during one of the CASP competitions, while the image on the right was created using traditional X-ray crystallography. And in looking at these, we can really see how close these two particular structures are. So let's take a look now at the NovaFold approach. And the NovaFold approach is this combination of both the threading and the ab initio methods. The NovaFold method will find structure fragments from multiple templates, build fragments not matching these templates, and then further refine the model as a whole. Because of the large computational power that this method necessitates, NovaFold uses the Amazon cloud. First, your query sequence is submitted on your desktop computer within Protein 3D. With your Amazon Web Services credentials, the sequence is then sent to a cluster of computers on the Amazon cloud, where the actual structure prediction computations take place. When complete, the predicted models will be returned to the desktop computer and Protein 3D for your analysis. So after all of this, how does NovaFold compare to iTaser? Right now, the results you get from the two approaches are very similar. However, with NovaFold, you do get our user-friendly interface, dedicated personal computer resources, so you don't have to wait to submit your prediction into a pipeline, the ability to run multiple predictions at one time, and our strong support staff to answer any questions that you have along the way. In the future, we will continue to improve this code with the collaboration of Dr. Zhang. Over time, DNASTAR hopes to improve the accuracy of the program even further, as well as Diverge to provide you with additional structure prediction analysis options. So now I'm going to go ahead and get into the software and open up Protein 3D here. Now, after we're familiarized with Protein 3D, we can then dive further into NovaFold. So to get started in Protein 3D, we can see the blank interface that I've opened up here. And you can very easily download any PDB structure file from the RSCB databank. So I'm going to go ahead and open up my web browser where I already have this RCSB protein databank opened up here. And I'm going to do a search for my protein of interest. So in this case, I'm going to go ahead and search for the 7-TIM structure here. Now, from PDB, I can go ahead and just download my files. And right now I can see that I have these couple PDB files here that I can choose to download, but any of these sequence files will work just as well within Protein 3D. So I've already downloaded these files to my desktop computer, so I'm going to go ahead and open up this application again. And so to open the file that we just downloaded from PDB, we'll select File, Open in Protein 3D. I'm going to select the 7-TIM structure file here. So we can now visualize this 3D protein structure within the structure view. Now, if we look at the top of the Protein 3D interface, if I go back up here, we can see several different options to look at our structure. So we can click and use the zoom tool here to zoom in and out of our structure of interest. We can use the move tool to slide our protein in and out and around the view. We can go ahead and we have two different rotate tools here to be able to rotate our structure any way we desire. And finally, if we decide we want to go back to the original view, we can go ahead and select the Reset Structure view here. Finally, we can also choose to click on any component of this particular interface to select it, and we can see that any residue that we select here in the protein structure in the structure view will then be displayed. So we can also very quickly and easily change the style of our protein as displayed in Protein 3D. So if we look to the bottom right of the interface, we can see a style panel here. Now, opening this panel, we can see many different tabs for customization here, including rendering, color, the sequence itself, or lighting for that particular structure. So I'm going to go ahead and expand this rendering tab as an example, and we can choose to display the backbone or the side chains of our particular protein in many different ways. So, for example, I want to display the backbone of the protein in ball and stick form. I can go ahead and do that here, as well as any of the other options that are available under this particular drop-down menu. In addition, we also have the option to alter our side chains for our particular structure that we're looking at. And we can select these in many different rendering forms as well. So we've examined the structure view now here in Protein 3D. So I'm going to go ahead and expand the sequence view so we can talk about that for a few minutes. Now, just below the structure view, we have the sequence view, which we can access through this button here. Now, if we open up this panel here, we can see that selections that are made at various points in the sequence view will correspondingly be highlighted in the structure view. So if I select any of the green helical elements, we can see that that will pop right up within the structure view above. If I select any of these red tube elements, then all of the residues involved in a particular binding site will be selected along with the binding site itself. Now, if we go back over to the right-hand side of the screen, we can look at our Explorer panel here. And we can see that if we expand down this Explorer panel, I'm going to expand it a little bit this way here. There we go. We can see all the different subunits and structural elements of that particular protein. So if we expand the Molecules tab, as we've already done here, we can see the two different subunits of this particular 7-TIM protein structure. So if we click on subunit A, we can see that that's correspondingly highlighted in both the structure and the sequence views that we looked at earlier. If we uncheck the box for this A subunit, then this particular subunit disappears from all coordinating structure and sequence views. So this complete integration between all views and panel selections is one of the really great things about the Protein 3D interface. So one of the other extremely important panels I'd like to talk about in Protein 3D is the Details panel. So if I select this here in the bottom right-hand corner of the screen, we can see that this displays important details about our protein of interest. So if we double-click on the background of the structure view, this will allow us to look at the details for the protein as a whole. Similarly, I can select any particular residue or subunit or structure of the protein and see the details on that as well. So here we can go ahead and take a quick look and see all the details for this particular 7-TIM structure. Now, one last view I'd like to discuss today is the Analysis view. So if I select that here and close down our sequence view a little bit to let us take a better look, we can see a list of applied methods to our particular structure. So if we go back up to this top right corner of the screen and we can expand the Methods panel here, And this will give us a list of all the particular methods that have been applied or could be applied to our particular structure. So we can really easily expand each of the methods' synopses over here in the analysis view. And we can look at individual method graphs as well as compare results across different applied methods. Additionally, under the Methods panel at the right, we can also expand the Parameters tab and select any of our methods, and we can choose to adjust our parameters as desired. So with all of the views and panels that we've discussed here today, Protean 3D has docking and rearrangement capabilities. So you can set up the Protean 3D interface to look however you'd like, customized for your particular workflows. So I can choose to detach this analysis view and look at it next to the structure view here. I can reattach things. I can also move any of these panels around to areas of interest. As you can see, I've just moved this details panel down to the bottom of the screen. So really any way you'd like to visualize your particular Protean 3D interface, you can set it up as such. So now after I've done a little work here in Protean 3D, I'll close down this particular example structure. I'd like to demonstrate to you our new application, NovaFold. So there are several different ways to access NovaFold, first of which you can select View, NovaFold here. Additionally, you can select File, Predict Structure with NovaFold, Structure, Predict Structures, Predict Structure with NovaFold. And then finally, if we go down to the bottom right corner of the screen, in the Details panel we discussed earlier, we can see that there is a Predict a Structure with NovaFold option. So I'm going to go ahead and select Predict a Structure with NovaFold here. And we can note that once we've opened up this NovaFold panel, which I'll expand to fill the screen a little more, this panel has the same docking capabilities as the other panels in Protean 3D that we've looked at. So you can, as we've done before, undock it, move it around, resize it, or reorganize the views any way that you want. So now that I've opened up this NovaFold panel, we have the option to look at the preview mode of NovaFold. Now, preview mode is enabled for users not currently logged into their DNASTAR accounts via the NovaFold panel. Now, this mode will show some pre-completed structure predictions, as we see these four below here, which allows you to view the results and give you an idea of what you might see as an output from running your own structure predictions. This is a good way, if you haven't already purchased NovaFold, to check out NovaFold a little bit and see what options are available. So I'm going to go ahead and log into NovaFold using a DNASTAR account here. And once I've successfully logged in, I can see the message from NovaFold telling me what my particular license information details. So currently I have 2 NovaFold licenses on this account. And again, you have flexible licensing options where you can purchase based on how many predictions you need, or purchase unlimited uses for a period of time, such as a week, a month, or a year. Now if we take a look at the toolbar here, we can see that we have several different options for the toolbar. But before I get into that, I do have a couple questions that came in here on Protean 3D and NovaFold. So I'll take a look at these. So the first question that we have here is someone noticed that I mentioned in the PowerPoint that additional programs come with the Structural Biology Suite, and what are those programs? So I'll just actually show you up here on the DNASTAR website. And once we've accessed that, we can go ahead under Products and select the Lasergene Structural Biology Suite, which is what Protean 3D, and NovaFold are all components of. If I open that up here, we've got this nice list of applications that then come with the purchase of the Structural Biology Suite. And again, we have Protean 3D, Protean, and NovaFold as we're discussing today. But in addition, we have a few other applications that come with this purchase for editing your sequence files and creating different publication quality graphics here. So I have another question here that asks if people can run multiple predictions at once. And if I go back to NovaFold here, yes, whatever you need. So we can set up your license to accommodate you for any of your particular projects. So, for example, I have 2 licenses on this account, as I mentioned earlier, which means that I can run 2 predictions at a time for as many times as I want until this particular account expires, which it lets you know right in this little message here. So this account will expire on May of next year. So for example, let's say you can also purchase predictions by the predictions. So if you purchase 10 predictions and you'd like to use these slowly over time, you might only need to run 1 prediction at a time. However, if you're down to the wire on a paper deadline, for example, you also may want to just run all of your predictions simultaneously, and we can set up your license that way as well. And then I have one more question here for this section about what happens if we stop the predictions. Do people get those predictions refunded? And yes, if you stop the prediction before it's completed and you have purchased a set number of predictions, so for example, if I've purchased 6 individual protein structure predictions within NovaFold, that prediction will be automatically refunded to your account, and you'll see that reflected in the upper right corner of the NovaFold panel with this information here. So that looks like all the questions that we have for right now, so I'm going to go ahead and continue to explain this NovaFold toolbar as we see in the upper left corner of the NovaFold panel. So here in this toolbar we have all the basic icons that are needed to manage your predictions. So from left to right, we have the option to create a prediction, which allows you to select your sequence files. And for each sequence file you select, an unsubmitted prediction will be created within the prediction list below. Next, we have the Run Prediction icon, and this officially starts running your prediction up on the Amazon cloud. Following that, we have the Terminate Prediction icon, and this will stop running your prediction and end the use of your cloud resources. And finally, we have the icon to delete a prediction. And this particular command will terminate your prediction and delete any of your results data that you have here. And this particular command is a permanent command. So once your data is deleted, there's no way of getting that back. So if we look below, we can see that we have the prediction list here. And any action of these commands in the toolbar will be reflected below within the prediction list. And the prediction list here summarizes all of the predictions associated with your DNASTAR NovaFold account. You can also have the option to sort this prediction list by clicking on any of the headers here at the top of the NovaFold panel. So I can sort by residue or the molecule itself, or when I submitted that particular molecule. So I'm going to go ahead and actually set up a prediction for us today. So I'm going to select the Create Prediction icon here, and then select the file I'd like to use. I'm going to use this FASTA file and open that up. And you can see that this new file has appeared now in the bottom corner of the prediction list here. This yellow icon that you see on the left will indicate this as an unsubmitted prediction. So I'm going to go ahead and click on this prediction here in the prediction list, and then select the Run Prediction icon. And here, this will open up the Run Prediction screen. Now, NovaFold will check to ensure in this step that your protein sequence fits within our 400 residue limit. While we are currently limiting our predictions to sequences 400 residues or less, we will be expanding soon as we optimize our algorithm for larger structures. Now, as we could see earlier from the prediction list, NovaFold does not offer the exact time that the cloud will take to process each prediction. However, we can estimate how long your prediction will take to run based on the number of residues in your sequence. So if we select this estimated cloud time link, we can take a look at the NovaFold benchmarks page on our DNASTAR website here. So in looking at this graph, we can see that for small proteins, which are about 100 residues or less, the NovaFold prediction will only take a matter of hours. However, for larger proteins, maybe 200 to 300 residues in length, the results may take a day or two to return. So this particular graph, we had NovaFold recreate the results of the Cas10 competition. And so the results from this recreation of the competition place it as very competitive with other members of that competition and give us those nice estimates of what our run times might be on the particular cloud. So if we close this window and go back to Protean 3D, we'll go ahead and click Run from the Run Predictions window. And if we give it a couple seconds here in the protein structure prediction list, we will see that it will begin to run right there. And so you can see that it says starting. And if we look at the icon over to the left, we can see this green triangle icon, which indicates that this prediction has started to run and is beginning to use cloud resources here. So while the prediction is running, I can safely close the NovaFold panel or Protean 3D and not impact this prediction at all. However, I will have to log back into my NovaFold account on Protean 3D if I want to check that this particular prediction is complete. So I'm going to go ahead and stop this prediction by selecting it and clicking the Terminate Prediction icon. And as we can see, the red icon here on the left has popped up showing that the prediction has been terminated. However, if our prediction had completed successfully and we had let it run for the entire length, we would see this nice little green check mark icon over here on the left that indicates that the prediction is complete. So after your prediction is complete, you have the option to select it from the prediction list and view the results in the Details panel. So I'm going to go ahead and expand this Details panel up and then select on a prediction that I've already run from it for us today. So we can see that we have several different tabs within this Details panel with different information related to our particular prediction. So one of the tabs I'd like to talk about is this Templates tab here that I'll go ahead and expand for us. So this Templates tab identifies the top 10 templates used in the threading approach discussed earlier. This tab displays the template and links to the PDB website. So if I click on any of these template links here, it'll take me right to that particular template entry on the PDB website page. So going back to Protean 3D here, we can also see that this particular tab will give us information on the coverage of each template, as well as the particular threading algorithm used to select that particular template. So the Results tab, as we can see here, is probably one of the most important tabs within the Details panel for looking at our structure prediction results here. So each model that we have displayed lists a C-score, a TM score, and an RMSD score. So the C-score, or the confidence score, will range from -5 to 2, with 2 being the best possible score available. The TM score will range from 0 to 1, with 1 being the best. Finally, the closer the RMSD, or the root mean squared deviation, is to 0, the better that is. So these models that you'll get in this Results tab are color-coded green, orange, or red for their particular predicted quality, with the coloring based on the model's TM score. Now, green models are the highest quality predicted models and are appropriate for drug design, drug screening, ligand docking, and molecular replacement workflows. The orange models are of a slightly lesser quality and are more appropriate for workflows such as mutagenesis design and predicting active sites, disease substitutions, alternative splicing patterns, and domain boundaries. Finally, the red models may be useful for predicting domain boundaries, but these are the least quality models that are available in your model list. To be qualified as a green model, the TM score threshold needs to be greater than 0.75. To be an orange model, the TM score needs to be between 0.5 and 0.75. And the red models, as discussed, will fall below this 0.5 TM score threshold. So if you click on the thumbnail of your model of interest, I'll click on this Model 1, as that is our best available model here. This will bring it up into the Protean 3D structure view for further analysis. So I'm going to go ahead and decrease the NovaFold tab to be able to expand the structure view here. And by housing NovaFold within Protean 3D, you do have the opportunity to take advantage of Protean 3D's graphical views and customization options that we did discuss earlier in the presentation. So any of the work changing rendering styles, changing coloring, applying methods that we discussed earlier in the presentation today, you do have the option to apply these to your NovaFold predictions. So I'm going to go ahead now and for the remainder of this presentation walk us through a few different workflows within Protean 3D. And while I won't use this particular result from our NovaFold structure prediction as a base for these two workflows that I'll show you, these workflows are applicable to any of your NovaFold structure prediction results. So I'm going to go ahead and close down this particular structure prediction result that we have here and open up that 7-TIM structure that we were working with earlier. Now, if we look at the methods panel here, if I expand that out for a second, and look at the analysis view, one of the most recently improved prediction methods is the DNASTAR Epitope prediction method, as we can see right here in our nice methods list that we have off to the right of the screen. I'm going to quickly walk you through how to use this epitope prediction method to view a solvent-accessible surface of the likely epitope in relation to the rest of the protein. Now, if you have an older version of Protean 3D, you may notice that this particular entry in the methods panel here has a Neoclone epitope prediction method as opposed to the DNASTAR epitope prediction method that we have listed now in our current 11.1 release. So in our most recent releases here, this improved prediction method will show more areas as possible epitopes than previous epitope prediction methods in the software did. So if I go ahead and go back to the structure view here, I'll get started with this particular workflow. And the first thing I'm going to want to do is open up the Surfaces panel, which I can do by selecting View, Explore, Surfaces. We can see that the Surfaces panel opened up right here in the side. So from the Surfaces panel, I'm going to want to select the Create a Surface icon, and we're going to go ahead and create a high-quality solvent-accessible surface for this workflow. We can see that this has selected the entire structure here and has now created a surface for the particular structure. So I'm going to go ahead and open up the Style panel here, expand that up a little bit so we can look at our options. And from the Style panel, I'm going to expand the Color tab. And I'm going to choose to change the color for our current selection, which is our particular surface that we have selected. So I'm going to go ahead and color this surface a nice blue color here and click OK. So yeah, we can see that now this surface has been colored blue. So if I unselect that surface and I go back to the Analysis tab here, I can see that we don't currently see any of the epitope prediction methods displayed here. So if I go up to this Right Methods panel and scroll down, I'm going to choose to check the box for the B-cell epitope prediction method here. So now if I look in the analysis view, I can see that this B-cell epitope prediction method is now displayed. So in the analysis view here, we can see that there are both antigenic regions, which are displayed in light pink, and highly antigenic models, which are displayed in the dark pink. And so we can expand this particular method for each subunit. And we can see confidence score graphs that allow us to analyze in more detail these particular epitope prediction regions. So in looking at these graphs, I might say that this particular region, this highly antigenic region with a high confidence score, I might think that this indicates a high probability that this would be a B-cell epitope. So I'm going to highlight this particular region of interest. And then I'm going to go ahead and go back to the Style panel with the Color tab here on the right and choose to color this selection a hot pink here. I'm going to go ahead and select OK. So after doing that now, and if I go back to the Structure view here by clicking on the Structure tab, I can then see that this particular highly antigenic region, the surface of which is now displayed in the hot pink that we chose to color. So we can now go through and analyze this particular structure model to visualize this region even better. So it looks like I do have one question in on the epitope prediction method here. And this question asks what the difference is between the highly antigenic and the antigenic regions. That's actually a really good question. This particular bioinformatics approach uses experimental antibody recognition data. And so for a set of neoclone peptides, the antigenicity of each peptide was determined by measuring the binding affinity of that peptide against various hybridoma-secreted antibodies in mice inoculated with that particular peptide. Now, highly antigenic regions are those in which at least 5% of these hybridoma cultures create antibodies that tightly bind the peptide. And the antigenic regions are regions in which at least 2.5% of which do so. So that about wraps it up for that epitope prediction workflow. One of the other workflows that's helpful to know in Protein 3D is how to open and align multiple structures within a single Protein 3D document. This will allow you to discover similarities and differences between the two structures. So to get this workflow started, I'm going to close out this epitope prediction workflow. And I've already downloaded two structures of interest from PDB for this workflow here. And so to open them in Protein 3D, I will select File, Open Group. And from this Open Group window, I'm going to choose to keep this Center on Screen to Align Similar Structures default setting checked. And I can now click the Add button and choose to add my two files. And so in this case, I want to add this one BFD file, and in addition, add this one PVD file here. So after I've opened both of those up here in the Open Group window, I can click OK. And now we can see that both of these structures are open in a single document. So if I go ahead to the Style panel here on the right and open up the Color tab, I can select the Color Style drop-down menu here. And what I'm going to want to select from this Style drop-down menu is the Color by Chain option. So now we can see that our one BFD structure consists of a single green chain, while our one PVD structure consists of both a blue and a yellow chain. So right now these two chains are unaligned, so they just overlap with each other randomly on the screen. And to align these particular chains, we will first go to the Explorer menu up at the top right of the panel. And we're going to want to go ahead and expand open the Molecules tab here. If I expand this all the way down, we can see all of our chains for these particular protein structures. Now, we're going to want to select the A chain from one BFD here, and then select the B chain from one PVD we'll just Control-click here and select both of those chains, or Command-click if you're on a Mac. So we can now align these selected chains here by going up to the top left of the screen and selecting Structure, Align Structures, and we'll select this rigid body alignment for selected regions here. And so now that the alignment is complete, we can go ahead and double-click on the background of the structure view here and open up the Details panel here in the bottom to analyze our aligned structures. So you can go ahead and see in this Details panel that it does give us an RMSD value for these aligned structures here. And so now we have the option to analyze that value for the two proteins to determine the degree of similarity between the structures. So as discussed with our NovaFold structure predictions, the closer our RMSD value is to 0, the better. So an RMSD value of less than 0.5 will indicate near identical structural similarity. RMSD from 0.5 to 2 will indicate still a very high degree of similarity. And an RMSD of 2 to 5 will indicate a lower but still noticeable degree of similarity. So from previous research on these particular structures, we already know that these two sequences have a 19% sequence similarity, as we can see from these two bottom chains overlapping here. So this corresponds really well with our end result of an RMSD value of 3.342, as we've gotten with this result here. So that about wraps it up then for this particular workflow within Protean 3D. And unless there are any other questions, that's about it for my presentation today. So I'm going to go ahead and bring us back to our DNASTAR website here. If you think of any other questions after the end of the webinar, you can feel free to email me at webinars@dnastar.com or tweet us at DNASTAR Inc. In addition, if you have any topic ideas for future webinars that you'd like to see, you can also send those to the same email. We have many different videos available on our website, as we can see under the Support tab. So feel free to check those out if you have some time. We have those here. We can see we have a nice list going for your protein analysis videos here. In addition, we have several different webinars coming down the pipeline. So if you look at our webinars page, we can see that we have 3 webinars coming up in August. So feel free to register for any of those webinars here. So thank you again for joining us today. We hope this webinar was helpful, and we hope to see you at a webinar soon.